Biological drawings provide a permanent record of what has been observed. In history and tradition, for hundreds of years since the 17th century, biologists have been accurately drawing what they are observing. Even today with digital photography, artists are often commissioned to come along and do an accurate representation of a biological drawing. Now, biological drawings will probably be one of the first PAGs you have to do, which is often a diagram of a heart, some drawing of some plant tissue, and a nucleus of a red blood cell. You will also be required to do them in the exam, and they're often very high mark questions, about 5 or 6 marks depending on the question. Not only do you need to be able to physically draw in the exam, you also need to be able to understand the rules associated with it. As you may do no drawing whatsoever, you just need to be able to say what should have been done better. So this is a good example of what a biological drawing will look like when you're finished. So let's dive into what we actually have to do. So take a look at this image here. Which one on the left hand side or the right hand side do you think is the better biological drawing? So it is the right hand side. This is the better biological drawing. And that's because there are two types of drawing. The first type is called a low power drawing. Is as it sounds. It is when the magnification is low, so think somewhere under about 100 magnification. That's not an exact number, but that's the idea. And in a low power drawing, you should not be drawing any individual cells. Instead, you are doing lining and layers of tissues. Now, it might mean that the drawing doesn't seem very scientific. It doesn't look like textbook diagrams. That's okay. The point of this is we are doing diagrams that look accurate and are a true reflection of what you have observed, so someone else could look at it and think that's what they would observe. We can then work out later on what we are observing, but the job of a biological drawing is to simply document what we're seeing. So with a low power drawing, it's layers of tissues within an organ that are completely circled. This one's been cut in half to show a comparison. Whereas high power drawings are more of probably what you think. We're drawing individual cells. Now you should only be doing a few cells, and the text on the screen is directly taken from the biological drawing handbook that I'll link in the description. Some key features when you do the biological drawing is it should be an outline, but we'll go into individual rules now. So this is a good example of a biological drawing with a few small mistakes, but overall it's pretty good. So the first key point, we should be drawing individual cells and going around the outside of them, and we should be avoiding drawing like this where the cells are crossing over, because in fact that cell doesn't cross over itself. The cell memory doesn't poke that way. So when you use your pencil, you should try your absolute hardest to try and make them as neat as possible without this little bit sticking out here. You should also, if there's no gaps between the cells, you should be avoiding gaps, and you should be avoiding shading. So we will run through the actual rules and describe what we mean by that. Use a sharp pencil. Now it should be sharp, there should be no thick lines. Often in the commentary from the exam board, they're discussing how they're not happy with thicker regions and thinner regions of the line. You might not necessarily be marked down for it, but that's what you should be aiming for all the time. You should be labeling all the relevant structures. Now, most of the time, those relevant structures are just going to be a couple of simple labels. It's unlikely that you'll ever have to do one quite complex as this in an exam. It's more likely that there are 3 labels you need to do, such as cytoplasm nucleus and cell membrane. You should be using a ruler for all the label lines, and you should not be adding arrowheads. You should be trying to make the proportions look like what you're observing, as the examiner will give you an image of a cell and you will be drawing that. Labels should be written horizontally, and the labeling lines should not be crossing. Now, the most common example of a failed biological drawing is a lack of scale bar and magnification. Now, to draw a scale bar, it's relatively straightforward because you should be measuring the thing that you are drawing. So if it's in a PAG, it's relatively straightforward. You measure it with your ruler, you put your straight line underneath, and you say however long that might be. Let's say 100 millimeters if you're drawing something that's quite large. In the exam, you will need to be given the magnification, or you need to calculate it within the question, and that should be documented too. So this is an example of a real exam question from a couple of years ago. So we're going to jump in here. So figure 21.2, so we identify that, that's this one here, is a high power image. So we immediately know now they're helping us in the question, but we are drawing individual cells, we're not drawing whole tissues. Whereas if we were asked to draw this one, this is an example of a tissue called an islet of Langerhans. So we would be drawing around the outside of that one. And we've been given space, so we would need to fill at least 50% of the space because we're given clear plain paper in the exam. Now this is the image close up. See if you can have a go at drawing 5 cells here and fully doing your biological drawing. Feel free to pause it on the rules to make sure you follow them. So this is the mark scheme for that question. This example here, we are going to mark together. This is given 3 marks. And I just want to highlight, if you're new to A-level biology, this part of the mark scheme is so incredibly valuable, and you should be going through every line and working out in your head, well, why is that given, why is that not given? You should do that every time before moving on. So this question here, we were given marking point 2, marking point 3, and marking point 6. So for marking point 6, they have mentioned the colour. That is an example of an annotation. So the difference between a label and annotation, a label is stating the name of what you're observing, and annotation is detail surrounding it. So you might say the cytoplasm, and then your annotation maybe, contains enzymes for metabolic reactions. They've also been given marking point 2 because they have drawn some cells to the correct proportions, and they've used over 50% of the page. If you're not super confident with your drawing, a biology drawing is not how good you can draw. It's how much you can follow the rules. They don't like gaps between the cells, and they really don't like overlapping lines between them. So you're just going to carefully be circling around what you're seeing. So here's another example now, and if you want to do a biology drawing and let me mark it for you, I'll do that absolutely free. If you do the biology drawing, if you then take a photo and email that to stemcelltuition@gmail.com, I will give you immediate feedback on how well you've done. Good luck with your biology drawings. It tends to come up really early in A-level biology, and next session we're going to go into the difference between magnification, resolution, and then applying it to different types of microscopy.